I'm going to talk a little bit about the newer fluorescent-based sensors for neurotransmitters and how, specifically how they relate to FSCV. And so these often are called genetically, oh, let's get the Y in the right spot, encoded sensors. I'm going to call them that. The ones that we'll start with are based on GPCRs, so G protein coupled receptors. And so most of your neurotransmitters, again, because I'm a neurotransmitter person, that's what I care about, have a GPCR. And so these are, again, typically 7 transmembrane, 1, 2, 3, 4, 5, 6, let's do one more. 7 transmembrane kind of proteins. And again, they can bind, they're already designed by nature to bind specifically different molecules. And so what they've done to these, and I'm sorry my green doesn't really work, so I'm going to change, is they've put on them some sort of GFP, and then specifically it's called permutated GFP there. And so when your molecule comes in and binds your GPCR, your receptor, what it's going to do when you have your dopamine that comes in to bind your receptor, what that's going to do again is change the conformation of your GFP, and now it's going to turn it on. And so you're going to get out, you're going to be able to do fluorescence. A reminder, all fluorescence, and we'll talk about the issues with fluorescence in a minute, but all fluorescence requires you to have some sort of excitation light, right? So, and then some sort of, you collect the emission. But yeah, you don't, it's not like you just go in a dark room and like, whoo, you know, it does this. It's like, oh no, you gotta put a light in to get a light out, kind of thing. So you gotta have a system that can do that. But the beauty of these is that they're basically taking a native receptor, slightly changing it, doing protein engineering to add on a tag. And so your body, you know, biology's in their favor, knows what to do with a protein that looks like this. And what it does with a protein that looks like this, right, is goes and, you know, puts these receptors on your surface. This is supposed to look like a neuron. You know, they could be presynaptic, they could be postsynaptic, but what you get is you get these receptors that are basically, now these are going to be not only receptors, right, but reporters out of the signal. And so as you know, take and dump out neurotransmitter, right, you know, as that kind of comes and it binds to the receptor, right, we'll turn it on, right, and this one will be turned on, etc., and you'll start to get fluorescence. And so these report, I wanted to do that very bad drawing, because I wanted to be clear that these are reporting from basically the cell surface and the neuron surface, right? So they are right at the source of where things are released. As receptors go, receptors are meant to bind. They're also meant to release back. You know, they don't bind forever. That wouldn't be very useful for you for cell signaling. So it'll bind, and as things are clear, you know, it'll unbind and it'll turn off again if the neurotransmitter is kind of cleared from the extracellular space. So the signals you get from these are, you know, neurotransmitter went up, neurotransmitter went down. You know, they can follow a transient signal. We'll put it that way. Okay, so if you have the right type of GPCR, they've now made these for a bunch of different things. I'll just list out some of them, but I'm sure I won't get them all. So for small molecules, they've made them for things like acetylcholine, dopamine, norepinephrine, serotonin, adenosine, ATP, histamine, and they're even trying to go into the realm of peptides. Peptides are always difficult because they are present in the brain at concentrations that are much lower than all of these other kind of small molecule neurotransmitters. Okay, so again, if you have a GPCR and you're a good protein engineer, you know, who's been trained in this, people, once they knew how to do one, they were able to go through and kind of do that and make a bunch. Okay, so there's something that's missing from the list. An astute neurochemist would realize that I did not write up two of the most famous kind of small molecule neurotransmitters, and that is because they don't have the right type of receptors. And so those ones that I missed, in case you're not such an astute neurochemist, would be glutamate and GABA. And they don't have the right type. They have ionotropic receptors rather than what they call metabotropic, you know, well, the ionotropic receptors are there. So they don't have the right kind of receptors that they can just go and like borrow nature's receptors. But Lauren Luger's group has gotten around this by using something called periplasmic binding proteins, and you'll never see that written out again. They call them PDPs. And so these come from bacteria, and again, so they've taken kind of a fake protein that will change conformations when bound to your glutamate or GABA, and then give you the fluorescence. And so he calls these things either like actually right to the table today. I-glue sniffers. I can't remember. Fluorescence is probably the FR. I don't remember. But he calls them like sniffers, and so he's made them for glutamate and for GABA. Just want the lowdown. Glutamate's much better than GABA. GABA is super hard. If you've watched this set of videos, how many times have I mentioned GABA? Almost never. Because we don't have good GABA sensors. So if you want to know in the future what you can do as a student to help the field, GABA sensors are really tough. But he did make one, but it's not nearly as sensitive as the glutamate one. Glutamate one has gone through a couple generations and is really good. So glutamate and GABA have to use a different approach. So not everything can use this approach. So for dopamine, for instance, the names of the sensors, Yulong Lee's group calls these grab sensors, and again, they'll use a subscript for whatever it is. Oh, it's the grab DA, and then oftentimes they'll put numbers or letters or that kind of thing after it to denote which type, because they'll make a bunch of different ones with different sensitivities, bindings, ranges, that kind of thing. And then Lin Tian's group has her own, I think they actually use a small d, which they call D-light. Kind of thing. And so that's just a different group making them. Sometimes they're based on different receptors, and that could be good or bad, depending on what your application is. So it might be, I think this one's D2 and that one's D1, but I might have them backwards. So you take a different receptor, it might give you different binding characteristics. Also, though, if you want to study that receptor, you might not want to mess with it, right? You know, so if you want to give drugs or something, any drug that will bind the native receptor will also block. You know, so if you want to do pharmacology, you got to be careful. You can't do pharmacology of the receptor that you've chosen as your sensor. I'll just put it that way. So having them for different receptors is actually useful. And as I said, we'll give you different profiles. So yeah, so these are some of the main ones in the field. You'll hear about the grab sensors. You'll hear about D-light. You know, and some of the other ones. Okay, so how do we get them in the cell? Again, they use kind of standard biology that's not super easy, I think, for us analytical chemists, if you're an electrochemist looking at this video. But in mammals, at least, they use viral vectors. Typically AAV, our lab has used something called Synvis virus. Excuse me. And so you put a gene encoding your sensor in there and get it to express in certain cells. That's my lesson on genetics today, not a real lot. But they'll inject these and wait. And again, biology does all the work for you. Biology, again, will kind of like get it to the right location, you know, and the cell surface and that kind of thing. In the fruit fly, we don't need viral vectors in the fruit fly. So if you do Drosophila, if you want to be a Drosophila person like the Benton lab does some Drosophila, here, again, they're more likely to do some sort of genetic breeding and make a stable line where this receptor grab will be always expressed in certain cells. Again, you can use genetics to express it in a certain type of cells. And so, yeah, so get to Drosophila and they're not using viral vectors instead. Okay, so what I wanted to talk about today, then, with that introduction, fine introduction to the viral vectors and the grab and D-light, etc. sensors, is just to kind of talk a little bit about some differences between genetically encoded sensors and FSCV. So we're going to call them genetically encoded sensors over here, and we'll put FSCV over here. And again, I am not an advocate that any technique is to try and figure out which one is best, right? But more what situations that you're going to run into, and what are the kind of advantages and disadvantages of each technique, because they all have them. So for the genetically encoded sensors, if you're going to say who wins on selectivity, again, I've been doing a series of videos today on different types of techniques, and FSCV hasn't won once. FSCV is good for quick and dirty things when you know what you're looking at, but direct electrochemistry isn't the best for selectivity. Here, we got nature working for us, right? We got a receptor that nature has highly tuned, oftentimes to be very selective for the target of interest. So you're going to get high selectivity there. And as I said, with FSCV, we're going to say it's not super selective, sadly. We can get some selectivity, but not great. These also tend to be pretty high sensitivity. Now again, it really depends on your friend the protein engineer and how good they are. As I said, they can change the binding pocket a little bit, the native receptor that they're working on. So it depends on protein engineering. I would say that FSCV has medium sensitivity. I don't know what that means really. It's not super high, but it's pretty good for what we want to see, but it's very molecule dependent. Right, so we're not going to be down in the weeds of looking at tiny, tiny concentrations, but it does depend on the molecule. So when we look at time response, for the grab sensors, here's someplace we have, like at least in our lab, detected a difference. They have very fast rise times, meaning as the sensor, if there's a change, they go up pretty quickly. And so they follow that pretty well. FSCV also has relatively fast rise times, but we have a problem of absorption, and that can sometimes slow things down a little bit. So the rise time for an FSCV is typically on the order of like a second. So it's maybe not instantaneous. I'm going to write my FSCV, I only really have two colors here, in orange, but FSCV is going to be pretty good. It's going to have a pretty fast rise time too. So rise time isn't the issue. If the signal goes up, the signal is going to go up. The issue becomes the fall off. And that fall off time can sometimes be slow. So for the, they're going to go up, and then, the signals say going to come back down. And that would be, again, we're drawing grab here in brown, and FSCV here in orange. And the FSCV, it turns out, is a lot faster. And so it's got a pretty rapid time, but it's got a very rapid off time. And so one of the things we've noticed is that the sensors sometimes the grab sensor tends to stay up longer than the FSCV when we're doing a simultaneous measurement. And so you say, why is that? And so I happened to be able to eat dinner once with Lauren Luger, who I told you made all those periplasmic binding, the sniffers. And he said, well, it's because we made them that way. He's like, if you want to get a good signal, a high fluorescence, you want it to hold on for a little while. You don't want it to bind, be done, right? So the higher the sensitivity they are, the slower they are. This is just a trade-off they make in protein engineering. If you don't mind it being lower sensitivity, they can make it faster. And so this is why when you go to buy these things or get them from different places, there's often, if you look in literature, like 5 different grab sensors. And you're like, for dopamine, you're like, why do we need 5? Well, there'll be different time responses, maybe different sensitivities, different ranges. They'll have ones that are super fast, but they won't be as sensitive, or ones that are more sensitive and then they'll be slower. And so I've come to the determination that the people who make these know this. The problem is I'm not sure all the people who use them know it. That's the problem, right? It gets translated down, they hand these out like candy to bunches of people who are, eager to use them. And it's good enough that you might think, well, that's got to be the real time course in the brain. And only when you compare it to something else, you go, oh, maybe that's not quite the real time course in the brain. Does it matter? I don't know. It depends on what you're looking at, right? What's your biological experiment? Maybe it being 2 seconds slow coming down isn't a big deal to you, right? You just wanted to know if it went up and down, or maybe magnitude, something like that. That kind of thing. So again, this is a platform, and so they've gone through, in the early years of this, they went through a wide variety of GPCRs. As I said, I think the periplasmic binding proteins are harder. It's definitely not as easy. But yeah, the people who could do this, went out there and, oh, what do I have a GPCR for? Oh, what can I turn it into be fluorescent? So it's a platform. And once you know how to use the platform, the nice thing is that you're always detecting GFP. So once you get a microscope, the emission and the excitation are the same if you switch in the histamine today and dopamine tomorrow and that kind of thing. It's a little bit of an issue. Now, and I would say over here, darn it, with FSCV, we're always limited to electroactive molecules. So we're limited to the ones that we can detect. If we can detect it really, we're usually really good at it, but we may not be good at it. So some of the issues with the fluorescent sensors include an issue that's always an issue in fluorescence. It's called photobleaching. And that is, if you leave your excitation signal on for a long time, eventually you'll stop getting fluorescence. You'll bleach your photofluor. And so this limits the time that you can collect an experiment for, and practically they don't usually leave their light on for more than 90 seconds. So this isn't the case where you can collect continuously, whereas with FSCV, people will often, turn on the waveform and go away and come back a couple hours later, right? You can collect continuously with FSCV and you don't have to worry. Where here, you got to turn it off and on. You got to get that protein time to, kind of rest in between detecting it for fluorescence. So it's not good for a continuous measurement. Other issues that can happen, so your body is set up to do receptor endocytosis, right? So how much receptor is expressed on a cell surface? Well, it can vary at different times, and people have tracked this, with different diseases and other things. Again, and so it can package that back into a little vesicle and get the receptors off. So how much is expressed is a really difficult thing, and it could change if there's something that's going on with endocytosis over the point of an experiment. Let's see. Let's also talk about calibration, and primarily that they don't. They don't do it here. And so with calibration, again, because the expression levels, every time you viral vector in, you get a different number of cells expressed, you really can't go from animal to animal and say, oh, I got fluorescent signal of X. That must mean this one has differences than I got Y in this other animal. They don't do that. They don't try and compare it because of different expression levels. Whereas with FSCV, right, we can do calibration and then back out, like change right current to concentration. And so one of the maddening things about reading fluorescence papers, if you're coming from an FSCV background, is they'll never tell you, ever, what micromolar concentration they think they're looking at. I finally found it in one paper, like, oh, maybe it's a micromolar. Like, they really don't know, right, because they don't try and calibrate like that. And so, I hope you can see this. Everything in this fluorescence is basically background subtracted and normalized. And don't worry, we background subtract too in FSCV. So I always thought that was one of our worst features, and these fluorescence ones haven't solved it. They're going to background subtract too. And so with background subtraction, again, if they get a signal like this, they're also going to take up a darker area and try and subtract for drift or that kind of thing. So they're going to do a background subtraction. And then the typical signal you get, again, is a normalized signal where it's the change in fluorescence over like kind of the total fluorescence. So delta F over F. That's going to be their kind of signal y-axis. Ours is current, right? And we can normalize if we want to, but that their signals are almost always normalized. And what you typically get is a few percentage. Like 1%, 2%. A really good sensor is 4%. Right? So these aren't huge changes in fluorescence, but the microscope can definitely pick them out, and you can see the difference. And so I think, again, that the fluorescence sensors are super useful because most biologists already knew how to do microscopy. The viral vectors aren't super scary for them, especially if they've been doing some stuff like optogenetics in mammals or things that already required that technology. But there are probably a few things that are, going to be limitations of just you can't do continuous experiments. You have to do some sort of stim. There's no way you're getting basal levels, right? You're not going to find out what the basal level of glutamate is in your slice. Darn it. It's just not going to happen. So like us, they're going to have to go do some sort of stimulation level. Like all things, they started in slices, and now they've moved in vivo. In vivo, it's usually called fiber photometry. And again, you need a fair amount of cells all releasing at once to get enough signal to see in vivo. But they'll put a fiber optic basically in the brain to detect the fluorescent signal in vivo. Sometimes they can make a little, like if you want the cortex, you could just put a little like window in the top of the brain too, but that only works if you want the cortex. It doesn't work if you want a deeper part in the brain, then you've got to go to implanting that. Oh, one last thing, and that is the number of colors. I'm running out of room, so I'm going to put it up here. I would say that, and as in a lot of things, they have basically two colors. Almost everything they start off making green, right, with green fluorescent protein, and then sometimes they work really hard to make red, and the red ones never are as sensitive as the greens due to some various optics kind of things. And so One of the reasons that in my lab I want to multiplex genetically encoded sensors with FSCV is neither are good for multiplexing, right? You're going to get one, maybe two molecules with FSCV at any time, you know, if it's the right molecules. You know, with here, you're going to have one, maybe two colors on the sensors, kind of thing. And so if you really want to look at what's happening in the brain with multiple neurotransmitters, neither of these are great in and of themselves. But if you can add them together, you should be able, they're similar enough in their responses and needing background subtraction and looking at, that kind of data that you should be able to kind of multiplex. And so that's where I personally think that, like as analytical chemists, we could add to the field by trying to multiplex different techniques, as well as just really studying their advantages and disadvantages and that